Hello everyone, I am Gianluca Lombardi from Sorbonne Université, Paris, and I will present to you my work about predicting binding affinity changes in protein-protein interaction with protein language models that I'm working on under the supervision of Alessandra Carbone. The study of the effects of mutations in protein-protein interaction is crucial for understanding the impact of genetic variations on protein function and disease. Meanwhile, the recent development of single-sequence protein language models has enabled us to exploit the exponential increase in protein sequencing data for large-scale prediction tasks. These models, based on a deep learning architecture called transformer, are able to generate context-aware encodings of residues in a protein, similarly to the semantic connection of words in a sentence. The generated encodings have been successfully adopted as input for downstream tasks such as secondary structure classification, protein location, and protein structure prediction. Our question then is the following. Can binding affinity changes in protein complexes be efficiently predicted by the new generation of protein language models? The goal would be the reconstruction of individual protein-protein interaction networks from protein sequences. Even single-point mutations can dramatically affect protein structure and, consequently, modify proteins' affinity with the different partners they interact with in order to perform their functions in their organism. For our work, we use different benchmark datasets extracted from SCMP database that contains more than 7,000 mutations for 345 complexes. For each entry, information is provided concerning the position and region of mutation in the complex, and binding affinities for wild-type and mutated complex. We focused in particular on three benchmark datasets: S1400s that contains single-point and multiple-point mutations, S1102 that contains the corresponding number of single-point mutations extracted from the first version of SCMP, and S487 that contains single-point mutations that were added in the second release of the database and are therefore used as test dataset. For the prediction of binding affinity variations, we developed a machine learning framework that can be divided in three main sections: the computation of protein encodings for wild types and mutated proteins using pre-trained protein language models. ESM2 gave the best results, but other pre-trained models were also tested. A Siamese convolutional light attention block that processes the encodings and gives as output a protein representation independent of the sequence length, and a regression block that combines protein representations into a single array and performs the prediction. The light attention block applies 1D convolutions of different kernel sizes to find weights for the amino acids that are then averaged over the sequence length. The module is therefore able to retrieve local information in the different regions of the protein. In contrast to standard attention, it has linear complexity with respect to sequence length, while still allowing for model interpretability. Before moving to the prediction results, an analysis of ESM2 embeddings was performed in relation to the mutations occurring in the protein. We restricted our analysis to single-point mutations and computed the correlation of different distance measures between the wild type and the mutated encodings. We found that for proteins that present many mutations, the average binding affinity variation per position correlates with the distance of embeddings difference from wild type. The trend can be also appreciated by projecting the embeddings differences into D with t-SNE reduction, where mutations are clustered by position. For model evaluation, 5-fold and 10-fold cross-validations were performed on S1400s and S1102 datasets respectively, as done in previous works. Here, we report the results with respect to Pearson correlation and root mean square error for the best version of the model up to date. The first plot shows that results are even higher for double and multiple mutations than for single point ones. We compare different language models to be used as backbones for embeddings generation, in particular ProtBERT, Protify, PROSI, and ESM2, with the latter giving the best results. Since we observe that interface residues play a crucial role in the variation of binding affinity, we fine-tune some of the pre-trained models on interface classification tasks, using sequences from ProteinNet 12 dataset for training, and LevelNet software for labels assignment. We also performed an evolutionary fine-tuning on proteins belonging to the most common species in SCMP, extracted from the string database. Results are reported in the table, showing an improvement in the model predictions, especially in smaller models, also with respect to the state-of-the-art method based on sequences, namely MuPiper. However, for the test dataset S487, our results are still slightly inferior, leaving room for potential improvements. Concerning interpretability, instead, we can visualize on the protein molecules the normalized attention weights, finding that for complexes with more accurate predictions, attention tends to focus on interface regions. Even if some noise is present, a similar behavior can be also appreciated in these two examples. Next steps will focus on improving the performance of the model, either by slightly modification of the architecture, especially in the combination of protein embeddings, or by trying to enrich the inputs with complex level embeddings or information extracted from multiple sequence alignments. We will test the model on other datasets and possibly also on other prediction tasks, given the flexibility of the framework. Another direction will be a more quantitative comparison of the attention maps extracted from different experiments to check their stability with respect to variations in the training sets. Thank you all for your attention, and if you're listening to this presentation before the conference, have a nice time in Lyon.